Dear conference participants, let me present a report to you. Proteolytic processes in organisms of different age rates exposed to xenoestrogens. The destruction of the endocrine system by chemicals, which are a group of compounds that negatively affect the endocrine system, is an organ problem of the day. Such substances are often found in everyday products and are epidemiologically associated with several diseases. Currently, the endocrine system biochemicals databank contains 1,000 molecules, including pesticides, natural and industrial products, cosmetics, medicine and food additives, and other low molecular weight xenobiotics. The environmental estrogens can be categorized in this group on these slides. Next slide. The Endocrine Society declares that endocrine disrupting chemicals are an exogenous chemical or mixture of chemicals and in the statement of principles. They interfere with any aspect of action of hormone and as an exogenous agent. One can be connected with the production, release, transport, metabolism, binding, action or elementation of natural hormones in the body. EDCs are responsible for the maintenance of homeostasis and the regulation of development processes. Next slide, please. Chemical bonds of xenoestrogens with plasma carrier protein have significantly lower affinity. In natural estrogens, this affinity is much better, so they are more easily accessible to target organs. Next slide, please. Age and estrogenic surroundings affected the difference in effects. A process known as developmental reprogramming can permanently reprogram normal physiological responses under the influence of changing environmental conditions. Such changes in physiological responses increase the body's susceptibility to diseases later in life. Next slide, please. The aim of study was determination of the effect of xenoestrogens on the proteolytic processes in different age rates. Next slide, please. Materials and methods. The experiments were conducted on Western rats exposed to exogenous estrogen for 45 days. At the beginning of the experiment, 3-month-old pubertal animals, group 2, and 6-month-old sexual mature rats, group 4, were involved. The control group consisted of intact, appropriated age animals, group 1 and 3. Next slide, please. Results and discussion. Cysteine cathepsines are important regulators and signaling molecules of many biological processes, some of these processes being listed below. Next slide, please. The enzymes are involved in the development of a number of pathological conditions. For example, cathepsines, which mechanically supports to show specific carcinogenicity of the like substance, caused invasion of tumor cells through the basement membrane. The activity of cysteine cathepsin L was reduced by 15% in the lower of pubertal families when compared with control, and 10% at the sexual mature individuals of group 4. Cathepsin B was activated by 13% respectively. Next slide, please. Table 1. Proteolytic indices of different age families rate exposed to elementary exogenous. Next slide, please. Table 1, Part 2. Next slide, please. Proteolytic inhibitors perform important physiological functions, delay the premature activation of proteolytic enzymes, protect proteolytic tissue from microbial enzymes, regulate the state of the coagulation system and fibrinolysis, affect arterial pressure and vascular permeability, apoptosis processes. Next slide, please. Exposure of the drug xenoestrogen in the brain of the rat in the pubertal period has been shown to activate cathepsin B by 10% compared with the control group of the same age. In the sexual mature females, the activity of the enzyme in the experimental group is reduced by 8%. Next slide, please. Widespread exposure of estrogens has led to the need for studies of biochemical changes in the kidneys. According to the results of studies of shifts in the proteolytic system, it has been found that trypsin activity increased by almost 40% in the kidneys of pubertal females and 8% in adult individuals. No significant difference were found for the alpha-1 AT index between group 3 and 4. For group 2, growth was 8%. A similar trend of change is characteristic from alpha-2 Mg. The activation was 5% and 17% respectively. Next slide, please. During the exposure of the xenoestrogen drug in the rat brain, both subjects underwent cathepsin L activation 7% group 2 and 30% group 4. Next slide, please. Indigenous intoxication is a clinical syndrome that arises at various pathological conditions due to the accumulation in tissue and biological fluid of the body of the metabolic destructive cell and tissue structures, destroyed protein molecules, accompanied by functional and morphological lesions of organs and body system. Next slide, please. The imbalance in the proteolytic system leads to excessive formation of the peptides with toxic properties. It has been shown their accumulations in the organs of rats of both experimental groups. Next slide, please. In summary, the eating food contaminated by exoestrogens led to changes in the proteolytic system and the development of endogenous intoxication. Next slide. Conclusions. Thus, the alimentary exposition of rats that exoestrogens leads to changes in the proteolytic system and the development of endogenous intoxication, which are organ-specific and age-dependent animals. The control difference of sign in an organism provides various influence on the following types of interaction, namely a cell and cell and extracellular matrix. In addition, soluble factors can be a trigger for disruption of information transmission by signaling pathways. We believe that the identified phenomena may lead to the suppression of one of the main mechanisms of removal of damaged cells from the population, named apoptosis. The information obtained can be a paradigm from the risk assessment and prevention of diseases, the etiology of which is the alimentary intake of xenoestrogens. Thank you for your attention.